So, hello students, and welcome to another session, live TA session for environmental biotechnology. My name is Ashutosh, and I am your TA for this course, environmental biotechnology. And today we are at week 10, so we'll be discussing some questions related to week 10 lectures, where Professor Pinaki sir has dealt with biodegradation and con- heavy metal contamination as well. Okay. So it's already 3:05. So let's start this class. So since we are at lecture week 10, so few more classes to go and we will finish all the lecture modules of this course, NPTEL course, Environmental Biotechnology. So I urge all of you to please participate, pay attention and interact with me. So these live session classes held on Saturday 3 p.m. are recorded classes. So whatever we will be discussing here, the questions as well as the recordings and the slide materials, everything will be shared to you through the NPTEL authorities, and a recording of this session will be available on YouTube for your referral. So you can always come back and listen to that for your explanation and clear your doubts. Okay, so this is the general set of instructions I give to all the participants every class, that please turn off your mic and video chat for smooth conduction of this session. Let me be the only one who is speaking to you throughout this presentation. You can interact with me via the chat box or the comment box in the YouTube section or the chat box in this live session. Please post your answers, your queries, or any doubts that you have or in concepts for me in the chat box. Okay, I'll be following it regularly for moving ahead in the presentation. So today, as we all know, we will be dealing with topics like bioremediation and heavy metal contamination of from lecture 40, 41, 42 and 43. Okay. So I have prepared 10 multiple choice questions which are similar to what you will receive in your assignments, live assignments that you need to submit for your exam, course and certification, and also in the final exam. And this live TA session is designed as such so as to familiarize with the pattern of the questions and many example questions, which is what we will solve in this session. Okay, with that much of an introduction, let's start this class. Let me share my slides with you and we can begin this presentation. I hope all the students can see my screen and hear me clearly. If yes, then just type yes in the chat box or give me a like or a thumbs up. I think we are clear to go to start this session. So once again, I welcome everyone to the live TA session for environmental biotechnology, where we will have a doubt clearing and a problem solving session. Practice problem week, lecture 10. So all the lectures covered in week 10 module of this course will be discussed today. So this course is taught by Professor Pinaki sir from IIT Kharagpur, while my name is Ashutosh. I am a PhD scholar at IIT Kanpur. And this course is brought to you by NPTEL, and I am a PMRF scholar who is dedicated to give teaching hours in this NPTEL live session. Okay, so this is the format of the presentation that we are going to address. There are 10 multiple choice questions, of which only one answer or option is the correct. I will pose the question and read the question aloud, read the options for you guys. You guys have to tell me what is the correct answer for this question. You can type your answer or options in the chat box, which we will collect, and I will look at the question, look back at the question and reveal what is the correct answer to this question. Then we will briefly understand the concept behind or the topic behind this question and why the correct answer is the correct answer. Okay, so this is the format that we are going to follow, and here is the first question of this session. Question number 1: dash maintains the bulk precursor pool and allows complete oxidation of the substrate. So there are some processes in the options. You have to tell me which of these processes maintains the bulk precursor pool and allows complete oxidation of the substrate. Option A, mineralization. Option B, assimilation. Option C, oxidative metabolism. Option D, central metabolism. Option E, secondary metabolism. Understood the question, and look at the options. Let's take a minute or two to collect some answers in the chat box and in the comment box for those who will be watching the recorded video. After a minute or so, I will come back to this question and tell you what is the correct answer to this question. Okay, I think it's time for coming back to this question to find out what is the correct answer. Let us check the chat box for what are the answers students have posted. Ashwini says option D. Okay. So the question is, which of these processes maintains the bulk precursor and allows complete oxidation of the substrate? Correct answer is Ashwini's option, which is option D, central metabolism. Correct answer is central metabolism. Central metabolism maintains the precursor pool and also involves in complete oxidation of the substrate. Now let us understand all other options that were presented to us. What is mineralization? What is assimilation? What is oxidative metabolism? What is secondary metabolism? And what is central metabolism? So what do we mean by mineralization? Mineralization is the complete breakdown of organic compounds to inorganic substances. So conversion of organic substances to inorganic substances, and mostly it happens by breakdown of organic substances. That means more complex organic substances are broken down into their individual component of inorganic substances, like breaking carbohydrates into minerals like nitrogen, carbon and hydrogen. Okay. And what is assimilation or immobilization? Uptake or incorporation of nutrients into the biomass is known as assimilation. uptake of the nutrients or minerals to create biomass, or incorporation into the biomass, is known as assimilation. So an example of mineralization versus immobilization: these organisms present in the soil, or microbes like some bugs and microbes and other plants, consume other organisms and excrete inorganic wastes and organic matter, which forms the pool of organic nitrogen, which undergoes mineralization to give us inorganic sources of nitrogen like ammonia and nitrates. These nutrients are usable by plants and are mobile in the soil. So, inorganic substances like ammonium, after mineralization, can be uptaken by plants and are mobile in the soil, but they get immobilized by organisms, retain nitrogen as the feed on organic matter, and grow, which is assimilation, and thus immobilize the nitrogen mineral back into organic nitrogen. So, this is a cycle between mineralization and assimilation or immobilization. These nutrients are stored in the soil organisms. Okay. Now, let us understand what is oxidative metabolism and what is secondary metabolism. Oxidative metabolism is nothing but cellular respiration by which cells use oxygen to break down food molecules like glucose to generate ATP, which is the energy currency of the cell. So, using oxygen to oxidize glucose to generate ATP. This is oxidative metabolism. So, here oxidative phosphorylation or cellular respiration happens in organelles like chloroplast and mitochondria. And what is secondary metabolism? Secondary metabolism involves pathways and production of secondary metabolites, which are not essential for organisms' survival but crucial for immediate survival, but crucial for long-term survival, adaptation, and ecological interactions, often synthesized from primary metabolites. So, whenever there is fixation of carbon source into the primary carbon compounds like phosphoenol pyruvate or 3-phosphoglycerate, these can undergo secondary metabolism, carbon metabolism, to give us the secondary metabolites, which might not be necessary for organisms' immediate survival but crucial for long term survival and ecological interactions. And many such compounds of secondary metabolites of plants are natural drugs and stimulants for the humans. Now, the correct answer is central metabolism. What was central metabolism? Core metabolic pathways like glycolysis, tricarboxylic acid cycle, and pentose phosphate pathway, which generate precursors as well as energy. So, the core metabolism pathway always produces precursor molecules like acetyl coenzyme A, alpha ketoglutarate, oxaloacetate, important precursor molecules which feed into the energy generating cycle, TCA cycle. And such pathways are glycolysis, TCA cycle, pentose phosphate pathway, which break down carbohydrates and other nutrients, producing ATP and reducing power, and also producing precursor metabolites like pyruvate, acetyl CoA, and alpha ketoglutarate. So, central metabolism is the pathway which preserves the precursor pool as well as undergoes oxidative phosphorylation, oxidative metabolism of the substrate, oxidative metabolism of the substrate to yield energy. That is why central metabolism is the correct answer in this question. I hope everyone is clear on the different definitions of all the options that were mentioned in the question. What is mineralization? What is assimilation? What we saw, what is oxidative phosphorylation? What is secondary metabolism? And what is central metabolism? If it is clear, then let me know in the comment box, and we can move ahead in the class. Okay, we will move ahead to the next question. Yes, thanks Ashwini. So, moving on to the second question. Question number 2. During biodegradation of a xenobiotic substance, which of these traits will make it degrade the quickest? And options are being structurally similar to biomolecules. Option B, having a linear carbon chain. Option C, having unsaturated carbon bonds. Option D, being an aromatic compound. And option B, being unsubstituted. Now you have to picture in mind what is a xenobiotic substance and what characteristics of a xenobiotic will make it degrade faster by microorganisms, of course. So here is the question and 5 options. Let me know what you think is your correct answer in the chat box or in the comment box. I will revisit this question in a minute or so. And then we will find out what is the correct answer to this question. Okay, I think it's enough time for question number 2. For some technical reason, I cannot see any answers. If Ashwini or some Priyanka has posted any answer, can you please post it again? I was just checking the comment box, but I lost all the previous texts. Else, I'll just move ahead with question number 2. During biodegradation of a xenobiotic substance, which of these traits will make it degrade the quickest? Correct answer to this question is option A. Being structurally similar to a biomolecule will enhance its degradation. Whereas we know that xenobiotic substance is not a biomolecule, right? Xenobiotic is a chemical substance. So what is a xenobiotic? Xenobiotics are chemical substances found within an organism that are not naturally produced by that organism or are present in an unusually high concentration. So these are xenobiotics found inside an organism but that are not naturally produced by the same organism. And there are many examples of xenobiotic compounds like industrial products, petrochemicals, oil gas mixtures. These are all chemicals which can be found in organisms. Heavy metals, aromatic and saturated hydrocarbons, nitrogen sulfur containing compounds, additives, preservatives, food industry, synthetic dyes like textile leather dyes, paints, anthraquinone, agricultural xenobiotic compounds like pesticides, fungicides, and weedicides, DDT, and chemical fertilizers, and medicinal pharmaceutical industry waste like antibiotics, beta blockers, X-ray media, non-steroidal anti-inflammatory drugs. So these are all examples of good classes of xenobiotic compounds, which are modified chemicals that can be found inside the living organism. Because we ingest it like pharmaceutical, we use it on our for growing our crops. We have petroleum and preservatives in our food. We have dyes and everything. We live around xenobiotic compounds. So the more similar a compound is to the naturally occurring biomolecule, the easier it is for microbes to recognize and metabolize it. So this is a key concept because the microbe has certain biomolecules which it can recognize, and most of these biodegradation are enzymatic and substrate reactions. So we know how enzymes are very specific to the structure of the substrate, and if the substrate structure is feasible and fitting inside the active site of the enzyme, then the enzyme will be able to recognize it better. So if it's a xenobiotic compound which is totally not organic or a biomolecule, then the best it can do is look similar to a naturally occurring biomolecule, which can be recognized by some of the enzymes or the microbe or the bacteria and metabolize it indeed. And other factors that generally increase the biodegradability includes simple linear structures, which was one of the options, presence of functional groups that enzyme can attack like OH or COH groups, sulfhydryl groups, lack of halogenation or bulky substitutions. So bulky substitution and heavy halogenation like bromo iodo derivatives are resistant to enzymatic attack because they have bulky groups. Unsaturation sometimes helps, but only in some cases because double bonds can be point of attack, but can also stabilize. And aromaticity and heavy substitution usually slows down degradation. So there are many factors which are affecting the biodegradation: chemical structure of the substrate, concentration of the substrate, what are the type of microbes that are present in the environment to degrade the substrate, what is the different composition of the microbial population, and what is the physico-chemical properties of the environment like temperature and the pH. So all these factors contribute to the biodegradability of a compound or any biological moiety or non-biological moiety. So here xenobiotics, when they are actually organic and inorganic chemicals which are not naturally present in the body or synthesized in the body. So xenobiotics degradation also depends on these factors, and when the most important factor, if it looks like one of the active biomolecules, then its recognition will be easier. Therefore option A was the correct answer, that xenobiotics, when they look similar to a biomolecule, then it is easier to biodegradation. Okay, is it clear to the students? If this is clear to the students, please let me know in the comment box or in the chat box with a yes or a thumbs up, and we can move ahead to the next question. Okay, coming to question number 3 is a problem based question. Antibiotic resistance is associated with frivolous consumption and improper disposal of antibiotics. Okay? However, antibiotic resistance genes have been detected in pristine habitats like Antarctica. They could have arisen because option A, all possible genes exist everywhere. Option B, antibiotics have existed in nature before humans. Option C, antibiotic resistance genes have alternate functions. And option D, antibiotics have simple structures. And option E, antibiotics are structurally similar to common substrates. So this is a 2 statement questions and 5 options. Read through your question carefully and remember it, and then you will be able to answer this question. Okay, let us take a minute or two and collect some answers for question number 3 in the comment and the chat box. And I will revisit this question later to reveal what is the correct answer to this question. Okay, I think it's time to revisit this question. This question is based on antibiotic resistance genes, and how does it how does it appear in places where, like pristine habitats like deep sea ice in Antarctica, if antibiotic resistance is associated with improper disposal of antibiotics. All right, so let us check the chat box for some answers. Ashwini has answered option B. Let us see what is the correct answer to this question. B is the correct answer. Ashwini is correct again. Antibiotics have existed in nature before humans. It's not just a human design concept, and antibiotic resistance did not appear randomly due to improper use of antibiotics, but they have been existing from ages even before humans existed. Okay, let us understand the concept of antibiotic resistance. Now, what is antibiotic resistance? Antibiotic resistance occurs when microbes, particularly bacteria, acquire antibiotic resistance genes, which are also known as ARGs, which are genetic elements that allow them to survive and multiply in the presence of antibiotics. So, antibiotics are kind of the selection pressure for a bacterial cell, which drives selection and evolution inside the bacterial genes. So, over time, these bacteria accumulate such antibiotic resistance or resistance to a particular chemical or a biochemical present in the environment, and such genes which offer resistance are known as ARGs or antibiotic resistance genes. These genes can spread through horizontal gene transfer, often via mobile genetic elements like plasmids present inside the bacteria, between different bacteria, including those found in humans, animals, and the environment. So, such antibiotic resistance genes are the main reason why many antibiotics are failing against new different varieties of bacteria, because bacteria are now evolving faster with more repertoire of antibiotic genes, and it's basically an arms race or a tug of war between the antibodies that we design or we have or we discover and the antibiotic resistance genes that the bacteria develops. So, in a natural habitat, antibiotic resistant genes are not new. Long before humans made antibiotics, microbes in soil produced antibiotics naturally, such as streptomyces. So, the first antibiotic that was to be discovered was penicillium, which was a naturally occurring fungus. Other microbes evolved resistance mechanism to survive a microbial arms race. Therefore, even remote pristine habitats can carry ARGs or antibiotic resistance genes because these genes predate human antibiotic use. So, such antibiotic resistance genes are not new, and they have been existing since decades, even before human activity has started or humans started developing antibiotics. So in a natural habitats, if there is a bacterium and there is a protist which consumes the bacteria for food, which eats up the bacteria. So this is called predation. The protist will try to eat the bacteria, this act of predation, but the bacteria will try to resist this in the act of defense. So bacteria can do this via two methods: antibiotic production, the bacteria can produce antibiotic, or maintenance, while the protist can evolve further to cancel out the antibiotics that are present in the bacteria via the horizontal, and also transfer these horizontally transfer these genes across population to evolve. So whatever antibiotic is presented to the bacteria over time, some of the few bacteria or colonies have mutations which confer advantage to certain bacteria against antibiotics, and these mutations in the genes now become the antibiotic resistance genes, and these are naturally present in the habitat and a part of bacterial evolution and speciation. Is it clear to the students? I hope it is. Just let me know in the chat box. We learnt about what are antibiotic resistance genes and how there is an arms race in which bacteria and other microbes develop antibiotic resistance and also develop antibiotics to avoid predation and to prolong their own generation and speciation. Yes, very good. Okay. Let's look at what is the fourth question of this session. Question number 4. Which of these nutrient biosynthetic product pairs is correct? Okay. So you have to identify what is the nutrient and what is the corresponding biosynthetic product the nutrient is involved in, and which is the correct match. Which is a match of the following question. Option A, SB amino acids. I hope you know what is SB as a metal stands for. B is magnesium, ATP and DNA. C is PO polonium for coenzymes. Calcium, option D, stabilizing the cell wall. And option E is LA or lanthanum, stabilizing ribosomes. For your information, SB is antimony or stibium. Which of these is a correct match for a nutrient and biosynthetic product? Okay. Take your time, look at all these matches and tell me what is the correct answer in the chat box. I will revisit this question in a minute or so. Okay, I think it's enough time. Coming back to this question, spent a lot of time on this. Which of these nutrient biosynthetic product pairs is correct? And Ashwini has answered option B. Let us see what is the correct answer. The correct answer is D, calcium stabilizing cell wall. Although B can also be a correct option, but there is a better option which can fit to ATP and DNA, which is phosphorus. If you remember the lectures from Professor Pinaki sir's uh, recorded lectures, he has mentioned phosphorus in ATP and DNA, which makes sense because adenosine triphosphate and both the DNA phosphate backbone is consisting of the nutrient phosphorus. Magnesium, although stabilizes ATP, ATP appears in magnesium complex, and magnesium is also important in DNA replication, and many other enzymes use magnesium as a cofactor. But the most fitting option is option D, calcium and stabilizing the cell wall. Okay, let us understand the role of calcium in bacterial or microbial cell wall. Calcium's main recognized role with bacterial envelopes is stabilizing the lipopolysaccharide layer of gram-negative bacteria. So here is a comparison between the cell walls of a gram-negative and a gram-positive bacteria. Gram-negative bacteria have an extra double membrane cell wall, an extracellular cell wall for consisting of lipopolysaccharide, the outer leaflet and the inner leaflet, which is bound by lipoprotein binding anchor to the peptidoglycan layer. Outside of the peptidoglycan layer, we have the lipopolysaccharide, and this lipopolysaccharide is stabilized or anchored into the outer membrane through calcium. While in the gram-positive bacteria, we have a thick layer of peptidoglycan and lipoteichoic acids. So that is why the gram stain cannot enter due to the presence of lipopolysaccharide into the gram-negative bacteria, whereas the peptidoglycan layer readily absorbs the gram stain, which distinguishes between gram-positive and gram-negative bacteria. In the outer membrane, the negatively charged phosphate groups of LPS repel one another. Divalent cations such as calcium and also magnesium, let me tell you, bridge these charges and cross-link adjacent LPS molecules, making the outer membrane more rigid and less permeable. So calcium conjugation with LPS in the membrane makes it more rigid and less permeable for the bacteria, such as strengthening the cell wall, giving additional strengthening properties to the cell wall offered by the calcium in gram-negative bacteria. And the other options in the elements: antimony, no known essential role in normal biochemistry, used in some drugs but not in biosynthesis. Magnesium is a cofactor for ATP utilizing enzymes, stabilizes ATP needed for ribosomes and nucleic acids. So magnesium is a complex, so it can be a correct answer. Polonium, highly radioactive, doesn't have a biological roles. Calcium is found in most cell walls, in plant cell wall as well. Cell signaling, stabilizing cell walls, especially gram-positive and gram-negative bacteria and algal cell walls. Lanthanum, not classically essential, but some methylotrophs use rare elements such as lanthanum for methylone dehydrogenase. So magnesium and calcium are both correct options, but calcium is more prevalent in terms of its action as a product, nutrient and biosynthetic product pair. And if you follow the lectures of Professor Pinaki, then he has also mentioned phosphorus with ATP and DNA as a nutrient biosynthetic product is a good match, is a better match than magnesium with ATP and DNA. Therefore, the correct answer is calcium with stabilizing the cell wall. I hope it is clear to the students. Yeah, we have spent a lot of time in question number 4. Let us move ahead to question number 5. A wastewater treatment plant with activated sludge system received waste containing high organic matter content and toluene. the level of toluene was determined to be non-toxic to bacteria. Okay, and after a few days, the organic matter was found to have partially degraded, while toluene content remained largely the same. This is most likely to have occurred because of dashed. Understood the question? In an activated sludge system, a mixture of organic matter and toluene was added, and it was determined that toluene was not toxic to the bacteria. But after a few days, when they saw it, when they checked the contents of the activated sludge, the organic matter has degraded but not toluene. What could be the possible explanation? Option A, lack of phosphorus. Option B, lack of nitrogen. Option C, presence of heavy metals. Option D, presence of high organic matter. Option E, toluene is not biodegradable. Which of these could be a reason that in a mixture of organic content, high organic content and toluene level remained largely the same, but the organic matter was partially degraded, where it has clearly been mentioned that toluene is non-toxic to these toluene levels was non-toxic to the bacteria. I hope you have understood the question. Go through the question once again yourself, look at the options and tell me what is the correct answer. Okay? I will revisit this question in a minute. Okay, let's look at this question again. This was the situation: wastewater treatment plant with activated sludge system. You added a waste containing high organic matter and toluene. The level of toluene was determined to be non-toxic, but after a few days you observed that organic matter has degraded but not the toluene. This is most likely because dashed. Okay, let us check the chat box. Ashwini has answered option E, the toluene is not biodegradable. Okay, let us see what is the correct answer to this question. Correct answer is option D, presence of high organic matter. In fact, toluene is totally biodegradable actually. Toluene is a hydrocarbon, right? Toluene is actually an aromatic hydrocarbon which can be biodegraded, and there are many such bacteria and strategies to biodegrade toluene as a hydrocarbon pollutant. But rather the answer here or the reason here is presence of high organic matter resulted in what we know as preferential substrate utilization or use. The bacteria preferentially used the organic matter as the substrate and then degraded it, switching back to toluene when the organic matter is completely degraded. Okay, let us understand what is this phenomena. Preferential substrate utilization is a microbial phenomena when microorganisms in a mixture of pollutants consume the most easily metabolizable compounds first before beginning to break down other less favorable compounds. Understood? This is the simplest definition. Preferential substrate utilization. The microbe has a preference for which substrate it can easily metabolize. And if it is present in abundance, which was the high organic matter content, then it starts degrading or consuming the most easily metabolizable compounds first. Therefore, the toluene levels did not see any change, but we rather saw changes in the organic matter levels. Now, there can be many reasons. Competitions for resources happens due to microbial preference. Many bacterial species can degrade toluene, but the enzymes required for this are often induced only when easier food sources are not available. So understand, there must be a selection pressure. When easier food sources are not available, only toluene as a food source is there, then the bacteria will activate those enzymes that are or induce those enzymes that can degrade toluene. In a wastewater plant where there is high organic matter, bacteria uses the readily available compounds for growth instead of switching to metabolic pathway for toluene. And there is also enzymatic competition. When both substrates are present, they may compete for same active sites on the microbial enzymes. This is what we also saw in the previous one. If the substrate doesn't fit the enzyme properly, then it will not likely be reduced or degraded or acted upon. The metabolic signals favor the degradation of more abundant organic matter. And toluene degradation pathways. While toluene is biodegradable, its degradation can be a complex process. For example, under anaerobic conditions, it requires specialized enzymes to initiate the process. In presence of high organic matter, these pathways are not activated because the bacteria are thriving on simpler compound. It's simple because it just got an alternative source, which is easier to digest and generate energy. That's why it is not touching toluene unless and until it uses up all the easy organic sources from the high organic matter content. So what really happens? Why the microbes degraded the organic matter but not the toluene? Even though the toluene level was non-toxic, both nitrogen and phosphorus are essential nutrients for microbial growth. But if either were lacking, both the organic matter and the toluene would show poor degradation. And not just toluenes. Heavy metals would inhibit degradation generally, but not selectively. And toluene is not biodegradable is just incorrect because toluene is biodegradable under aerobic conditions, though the process is slow. So when microbes are offered an easily degradable carbon source like general organic matter, they will prefer that over a more recalcitrant compound such as toluene. This is called catabolite repression or substrate preference, with what we call preferential substrate utilization. As a result, the more complex or less preferred compound remains largely unchanged until the simple organics are depleted and the bacteria switches the metabolic pathways. So in the activated sludge, if we do a mixture of organic matter and toluene, because of the presence of high organic matter, the first outcome would be degradation of organic matter, but the toluene levels will remain the same. So this is what was happening in the question. I hope we have all cleared our concepts and understood this. If yes, then we can move ahead to the next question. The preference substrate utilization. Okay, cool. Now here is the sixth question. Biosurfactants primarily help in the degradation of dash. This can be a pretty easy question. Biosurfactants primarily help in the degradation of dash. Option A, heavy metals. Option B, insoluble non-polar molecules. Option C, soluble non-polar molecules. Option D, insoluble polar molecules. Option E, soluble polar molecules. Where would you use biosurfactants? And they primarily help or are designed to target which class of pollutants? Okay, let's wait for a minute and collect some answers. I'll be quickly back to this question. Okay, I think it's time to revisit this question. It's already a few minutes now. Biosurfactants primarily help in the degradation of dash, and in the chat box, option B. Okay, so let us check what is the correct answer to this question. Indeed, option B is the correct answer. Biosurfactants are primarily used for degradation of insoluble non-polar pollutants. Non-polar means hydrophobic and they are insoluble. Okay, what are biosurfactants? The term biosurfactants is a conjugation of two words, bio, which is derived from a biological source like bacteria or fungus, but it's a surfactant, which are compounds that lower the surface tension between two liquids. So, surfactant has this bodily composition. One part of it is a hydrophilic moiety, and the other part is a hydrophobic. So, this will allow or decrease the surface tension between two liquids and thus help separate each other. Now, the main role of biosurfactants is to reduce surface and interfacial tension, increasing the solubility and bioavailability of hydrophobic or non polar compounds in the water. This allows microbes to access and degrade substances that would otherwise stay separated from the aqueous phase. Very important. Just like hydrocarbons, which are totally hydrophobic, non polar compounds, biosurfactants are mainly largely used for treating oil spills and hydrocarbon contamination. So these are microbes which can produce these biosurfactant and enzymes, which has two parts constitutively, an hydrophilic part and a hydrophobic part, an enzyme. When added to a hydrophobic pollutant, it can lead to its biodegradation via two mechanisms: formation of micelles and incorporation of the pollutant inside the cell of the microbe, and attachment of the microbial cell to the pollutant, such that then the cell can reduce or act upon the degradation of the pollutant. Because biosurfactants emulsify and solubilize hydrophobic water insoluble compounds such as oils, hydrocarbons, and some xenobiotics, enhancing their microbial degradation. So, such biosurfactants are useful in enhancing the microbial degradation of hydrophobic pollutants like oils and hydrocarbons, and some xenobiotics and phenolic compounds, and so on and so forth. I hope it is easy to understand right now. You have understood this question? Okay. Let us move to the next question, question number 7. Which of the following is a viable strategy to deal with toxic heavy metals? Which of the following is a viable strategy to deal with toxic heavy metals? Option A, complete oxidation. Option B, dissolution. Option C, precipitation. Option D, mineralization. And option E, dilution. A viable strategy among all these which is used to deal with toxic heavy metals. Okay, so this is the question. Read through the options and tell me what you think is the correct answer in the chat box or in the comment box. Okay, I think it's time to revisit this question. Let us check what are the answers in the comment box. Which of the following strategy is viable for dealing with toxic heavy metals? Ashwini has answered option C and Tuhin is saying option C. Okay. Correct answer to this question is option C. Both Ashwini and Tuhin are correct. Well done. Precipitation method, precipitation by microorganisms, is the correct strategy to deal with toxic heavy metals. Okay. Let us understand this procedure. Heavy metals have certain characteristic properties that they cannot be destroyed or oxidized into harmless forms the way organic compounds can. Organic compounds can be oxidized into harmless forms, but mostly heavy metals cannot be, because they are more heavy metals are mostly unreactive. Instead, remediation strategies focus on immobilizing or removing them. So, if you reduce the flow or immobilize the heavy metals or remove them from the particular environment, then that is the only method that can be used. And one of the most effective methods in converting soluble toxic metal ions into insoluble precipitates such as sulfides, hydroxides, or phosphates. This is one of the most feasible method. This reduces their bioavailability and toxicity and allows easier removal from water or sludge. So, reducing or precipitating heavy metals as their salts, such as sulfides, hydroxide, or phosphate, into insoluble precipitates of these heavy metals are one of the most effective methods to reduce their ionic bioavailability and toxicity and to allow easier removal from the water or the sludge. So, heavy metals can come from many sources like industry and urbanization, and these are some of the examples from heavy metals: cadmium, nickel, mercury, arsenic, and copper and lead. These are many of the heavy metals. These can cause contamination of water, bioaccumulation in bigger life forms, and biomagnification, and pose damage to environmental and life forms. So, for remediation of heavy metals, we utilize living organisms such as plant, fungus, biofilms, bacteria, or algae to aid in precipitation and reduction of such heavy metals through various processes like bioabsorption, bioaccumulation, biomineralization, bioleaching, bioprecipitation, and biotransformation to make these heavy metal varieties safe to earth and efficient remediation, and also a very feasible process. So, in these ways, many of the microorganisms can be utilized for heavy metal bioremediation, detoxification, extracellular chelation, binding to the cell wall components. So, heavy metals can bind to the cell wall components and the transport channels, and intracellular chelation as well. So, chelation in the sense satisfying the charge or the oxidation state of the metal and precipitating it as complexes. Precipitation, adsorption, metal efflux through chelation, these are bioaccumulation strategies. Biosurfactants for bioleaching, biotransformation, oxidation or reduction of such heavy metals, hydrolysis and condensation, bioabsorption or biosorption, microprecipitation, ion exchange, ligand exchange, and many other methods of bioprecipitation. So, heavy metals cannot be oxidized, and the only way to get rid of them is via immobilizing them through precipitation strategies, or which is known as biotransformation or precipitation. Okay, moving on to the next question, question number 8. Toxicity of metals is mostly due to their affinity for the dash groups of proteins. The question is, why are heavy metals toxic to proteins? They react with certain groups of proteins which make them really toxic. Option A, methyl group. Option B, formyl group. Option C, carbonyl group. Option D, amine group. Option E, sulfhydryl group. Which groups of the proteins are easily recognized by metals or heavy metals, and reaction with them can cause toxicity in those proteins? The question is placed before you. Take some time, think about the answer, and tell me in the chat box or in the comment section what do you think is the correct answer to this question. Okay, it's enough time for question number 8. Coming back to find out what is the correct answer. Toxicity of metals is mostly due to their affinity for dash groups of proteins. Checking the comment box, Ashwini and Tuhin are consensusly saying option E, sulfhydryl. Okay? Correct answer to this question is sulfhydryl groups. This is the correct answer. Again, you guys have answered correctly. Sulfhydryl groups of the protein is what is reactive to most of the heavy metals, which also reduces the enzyme, inactivates enzymatic activity and disrupts the structure of the such proteins. Now, what are these sulfhydryl groups? Let us understand. Toxicity of most heavy metals come from their strong tendency to bind to sulfhydryl groups or thiol groups, what we say. Many enzymes and structural proteins rely on cysteine residues which contain sulfhydryl groups for their activity or stability. When metals such as mercury, cadmium, or lead bind to these SH groups, they disrupt the protein structure, inactivate enzyme, and interfere with cellular processes. So, here is an example. protein or enzyme, the amino acids which contains these kind of groups, the sulfur group, are two such amino acids. One is the cysteine amino acid and one is the methyl alanine. Methyl, sorry, methionine. Methionine and cysteine are the two amino acids which have sulfur groups. But most frequently, cysteine is found in the active sites and also is important for formation of disulfide bonds which bridge protein structures and domains together. So when a protein or enzyme has exposed cysteine residue sulfhydryl groups, it can react with a divalent heavy metal ion to form a complex where the reactive sulfhydryl group is now sequestered, changing the protein structure and disrupting enzymatic active sites. Two molecules of methylated protein or enzyme with methylated sulfhydryl groups can also react with heavy metals, forming double complexes, thus deactivating or aggregating proteins as well. Therefore, the toxicity of heavy metals comes through its reactivity towards these thiol or sulfhydryl groups. Heavy metals such as mercury, lead, and cadmium or arsenic are highly attracted to the thiol side chain of cysteine residues. By binding to these groups, metal exert their effects in several ways. Like inhibiting enzymes, many enzymes depend on free thiol groups in their active sites to function properly. When toxic metals bind to these sites, they inactivate the enzyme, disrupting vital metabolic pathways. Okay, so enzymatic active sites contains these thiol groups. Disruption of protein structure. The binding of heavy metals can cause a protein's three-dimensional structure to collapse, leading to denaturation. This can unfold the protein, expose hydrophobic regions, and cause the protein to aggregate, ultimately leading to loss of function of the protein. And many heavy metals are also known to deplete antioxidants because cysteine is a key component of glutathione, which is a major antioxidant in the body, which leads to increase in oxidative stress or increase in the levels of reactive oxygen species. By reacting with thiol groups in the glutathione, toxic metals deplete the cell's antioxidant defenses. This can lead to oxidative stress, which further damages cellular components like electron transport system damage, DNA damage directly, mitochondrial damage, protein aggregation and damage, and membrane disruption. That is how heavy metals can cause such wide range of toxicity, and they are also very toxic to humans. They can cause kidney failure, neurotoxicity, and many other skin infection and diseases. So the protein sulfhydryl groups are the main target of these heavy metals. I hope it is clear to the students. If you have well understood this, then let me know in the chat box, and then we can move ahead to the next question. Okay, let's move to the next question and look at it. Question number 9. Which of the following processes tends to yield energy? Okay, which of the following processes can yield energy? Option A, bioaccumulation. Option B, biosorption. Option C, bioreduction. Option D, biomineralization. Option E, bioleaching. These are all the methods in which a microbial cell helps in remediation. For your hint, now which of these remediation processes tends to yield energy in a microbial cell? Let's take a time and post our answers in the chat box, and I will be back to this question for revealing what is the correct answer. Okay, I think it's time to return back to this question. Let us check what are the answers in the comment box. Tuhin has answered option C. Okay. Which of the following processes tends to yield energy? The correct answer is option C, bioreduction. Well done, Tuhin. Bioreduction is the correct answer. Now, what is bioreduction? Microorganisms can use certain metals or compounds as electron acceptors in their respiratory chains under anaerobic conditions. And bioreduction of heavy metals work on the same principle as the iron reduction example in the schematic. Microbes use the metal ions as electron acceptors during respiration. So microbes can use these heavy metal ions as electron acceptors and then reduce them in the process, thus producing energy. This changes the oxidation state of the metal, often making it less soluble, less mobile, and less toxic. While other options like bioaccumulation and biosorption are mainly passive uptake or binding, they do not release energy. Biomineralization is often formation of the minerals from biological processes, often energy neutral or consuming energy, and bioleaching involves solubilizing metals via oxidation but does not directly yield energy for the microbe in most cases. So the correct answer is bioreduction because it is directly linked to electron acceptor in the electron transport chain to microbial respiration and can be a source of metabolic energy. So looking at all the processes at once for metal microorganism interactions, it can be bioabsorption or biosorption, bioleaching on the extracellular side, insoluble metal plus organic acid to soluble metal acid complexes. Biotransformation can be enzymatic reduction of soluble to insoluble precipitate. Bioaccumulation or accumulating inside the microbe, inside the cell of the microbe, and biomineralization, converting or oxidizing the metal ion into mineral deposits. Biotransformation of heavy metals to biological processes that alter the chemical form, oxidation state, solubility or toxicity of metals without actually destroying the element. And microorganisms accomplish this via enzymatic reactions indirectly through metabolic byproducts. Bioreduction, reducing metals to a lower oxidation state such as uranium 6 to uranium 4, usually making them less soluble and less toxic, and oxidizing metals to a higher oxidation state is biooxidation such as in arsenic, which can sometimes immobilize or mobilize the metal. Okay, so here what the concept that we learnt is in bioreduction the heavy metal can be used as an electron acceptor and thus connecting it to the energy yielding process directly of the microorganism. I hope it is clear to the students. If yes, then let me know in the comment box that you have understood, and then we can move to the next question. Very good. So now looking at the last question of the session, question number 10. Question number 10 is, which of the following is regarded as the simplest way to remove toxic metals? Simplest way to remove toxic metals. Option A, bioaccumulation. Option B, biosorption. Option C, bioreduction. Option D, biomineralization. Option E, bioleaching. Which one of these processes is regarded as the simplest way to remove toxic metals via the microwave pathway? Take your time to tell me what is the correct answer in the comment box, and I will visit this question again to reveal what is the final correct answer for question 10. Okay, I think it is time to revisit this question. Which of the following is regarded as the simplest way to remove toxic metals? Let us check the comment section for answers. Tuhin has answered option A. Okay. Let us see what is the correct answer to this question. Correct answer to question number 10 is option B, biosorption. Biosorption is the simplest way to remove toxic metals. Simplest, most cost effective, and while not harming the microbe as well. So bioaccumulation is also a simple method, but I will tell you why option B is a better choice. Okay? Now let us understand biosorption. The simplest way to remove any toxic metal with microbes is through biosorption, which is a passive process where metal ions bind to the surface of the live or dead microbial biomass without needing cell metabolism. So this is the major factor. Even a live or a dead cell can bind to the microbial, can be used for bioabsorption, because in bioabsorption or absorption of the toxic metal, it immobilizes it on the surface of the dead or alive biomass of the microbiome. Metal binding: microbes are the functional groups on their cell walls that attract and bind metal ions. It is as simple as that. And what are the mechanisms? This can be through electrostatic interactions, like positive charge metal ions attracted to negatively charged membranes or proteins. Surface complexation: metals form chemical bonds with specific functional groups on the cells. Or precipitation: metals can react with components on the cell surface to form insoluble precipitates that remain attached to the biomass. And it is also a passive process. Unlike other forms of bioremediation, bioabsorption doesn't require the microbe to be metabolically active or alive. So, this is the only difference between bioaccumulation and bioadsorption. In accumulation, the microbe has to actively pump or intake the mineral and accumulate inside a living microorganisms. So, for bioabsorption, we do not need the microorganism to be alive as well. It's a passive process. The conjugation or interaction of the heavy metal with the cell membrane or cell wall of the bacteria is enough for bioabsorption. Dead biomass or cellular fragments can also be used, and the microbe need not be metabolically active or dead for bioabsorption. The bioabsorption method is effective because it often inexpensive, can use readily available biomass, and allows for selective removal by choosing appropriate microbes. The process involves electrostatic interaction, as we just saw, complexation or precipitation, making the toxic metals less mobile and easier to manage. And these are other several advantages of bioabsorption that it offers: no bioaccumulation, removal and recovery of the component, no toxicity limits for heavy metals, low cost and easily available, which is also eco-friendly. These metals can be regenerated and reused because there is no change in chemical composition of the metal. Generation of minimum secondary waste and high efficiency, high surface area to volume ratio. These are some of the advantages of bioabsorption, which makes it the simplest and most affordable technique for ridding the environment of toxic heavy metals. I hope it is clear to the students that why bioabsorption is the best method or easiest method, simplest method for removing heavy metal toxicity. I hope it is. And so with this, yes, thanks to him. So with this, we have come to the end of this problem solving session. I will end my slideshow and come back to the call again. Yeah. So with this, we have come to the end of this session. We have encountered 10 such questions from lecture week 10 where we dealt with bioremediation and heavy metal contamination. These were the topics that were discovered that were discussed by Professor Pinaki in all his lectures. So it was happy to conduct this class, and I hope you guys could learn something about the questions and about the brief concepts that we discussed alongside these questions. If you have any doubt or you need further understanding, please feel free to interrupt me through the class and post your questions on the chat box. And if not, then I will end this class and see you next week with lecture week 11. Be prepared, go through your lectures and try to participate in this class to make the most out of it. I hope you liked the presentation. If you have any suggestions in the teaching style or in the presentation or in my that will help in my improvement, you are free to give me suggestions. And thank you for joining this class. I will end this call. Let's meet next Saturday at the same time, 3 p.m., to discuss lecture week 7. Okay? I will end this call. Thank you for joining.